أعزائي طلاب المرحلة الرابعة السلام عليكم ورحمة الله وبركاته. We discussed in the previous lectures the chemotherapy, and we started with this group, the antimetabolites here, and we talked about methotrexate and other members. Then we talked about the mechanism of action, side effects, etc. Today we will continue in this series, and we will talk about 6-mercaptopurine, also another member in antimetabolites. And this drug is considered as a purine antimetabolite. And there is another drug is also closely related to mercaptopurine. It is called azathioprine. It is an immunosuppressant drug and exerts its cytotoxic effect after conversion to 6-MP, 6-mercaptopurine. 6-MP is now primarily used for maintenance of remission in acute lymphoblastic leukemia. This is the major use for this drug. And both of them are useful in the treatment of other diseases such as Crohn's disease, in addition to anti-cancer effect. Then we have fludarabine, and fludarabine is a purine analog. So both of them, let's say, purine antimetabolite. This one is useful in the treatment of chronic lymphocytic leukemia, similar to the 6-MP hair cell leukemia and non-Hodgkin lymphoma. So it has more clinical practices. Fludarabine is a prodrug, and when it moves inside the cells, it like in the process of transferring of this drug, there is a kind of transformation by removing the phosphate group from this drug and converted it into a metabolite called 2-FRA. And this one is taken by cells, and inside the cells there is another process of phosphorylation, adding a phosphate group to the molecule of the drug. And this step is done by deoxycytidine kinase, a kinase enzyme. Then the cytotoxic effect is related to the phosphorylated metabolite inside the cells. Even this mechanism is not clearly understood. However, the triphosphate metabolite of the drug is incorporated into DNA and RNA and decreases the synthesis both of them in the S phase and affected on their function. So it means like if it is incorporated into DNA, there is no more transcription to RNA, and if it is incorporated to RNA, there is no more synthesis for the proteins. There are a couple of mechanisms that are responsible for the resistance to this drug, such as reduced uptake into cells or lack of deoxycytidine kinase. There is no conversion again to the phosphorylated metabolite, or decrease the affinity. Fludarabine is administered either intravenously, parenteral administration, or orally. Because the intestinal flora, this drug is split of the intestinal flora, split of the sugar group, and yield or generate a very toxic metabolite called fluoroadenine. Then we have 5-FU or 5-fluorouracil. This one is a pyrimidine analog, unlike the two previous drugs. And in the structures, there is a place for the hydrogen atom in the position 5 in the uracil ring, and is replaced by a fluoride molecule. Also, the fluorine interferes with they added the fluorine to this molecule, interferes with the conversion of deoxyuridylic acid to thymidyllic acid. And we will see this in the last step right here. So in general, this drug causes a deprivation of cell thymidine, and thymidine it is an essential precursor for DNA synthesis. It is an essential building unit for DNA synthesis. There is no more thymidine inside the cells. 5-FU is used primarily for slow ground solid tumors and also can be used topically in case of superficial basal cell carcinomas. So with the mechanism of action, 5-FU, it has no antineoplastic activity. However, when it enters inside the, like here it is outside the cell, then it is its presence inside the cells, and here it is converted by sequential steps by different kinases and enzymes. It is converted finally to what it is called 5-FDUMP, and it means 5-fluorodeoxyuridine monophosphate. And this one, it is a competitive inhibitor with DUMP, deoxyuridine monophosphate for thymidylase synthase. So it inhibits thymidylase synthase. So there is no more conversion to thymidylene or thymidine, and this step causes deprivation of thymidine, and it is an essential building unit for DNA. So this is the same thing right here, and DNA synthesis at the end or as a net result decreases due to the lack of thymidine, and this leading to the imbalanced cell growth and thymidine-less death of rapidly dividing cells. So death of carcinoma cells due to thymidine deprivation. Another mechanism is also related to 5-FU. It is incorporated into RNA, as we can see here from these side steps, but also it is incorporated into DNA. And incorporation of this molecule into DNA, so it is like a molecule of thymidine with a fluorine side group. This causes damage to the DNA, so it prevents the elongation of DNA, and there is no more synthesis for DNA strands. And this causes the anti-cancer effects of 5-FU, and this effect is more specific to the S phase of the cell cycle. With the pharmacokinetic, because of the severe toxicity that is related to 5-FU, 5-FU is administered by intraparenteral route. Also, as we said in the previous slide, this drug can be administered topically in case of the skin cancer. Also, this drug penetrates very well to all tissues, including CNS, so it can be used for tumors in the brain. But it is rapidly metabolized in liver, lungs, and kidneys. It is eventually, after metabolism, converted to a metabolite called fluorobeta-alanine, which is removed in the urine with the unchanged parent drug. Here we have an important phenomenon. It is related to a certain enzyme. It is called dihydropyrimidine dehydrogenase, or DPD, and this one is responsible for the catabolism of 5-FU and decreases its bioavailability. The problem with this enzyme, its level could be differ from patient to patient in around 6-fold. So it is a huge difference in the concentrations, and this leads to affects too much on the catabolism or metabolism of these drugs. So some patients with the same doses could have 6-fold more in the concentration of 5-FU, and this is of course responsible for more side effects and more toxic effects such as pancytopenia, mucositis, life-threatening diarrhea. So it is important to assess DPD during using 5-FU. Then we have capecitabine, and capecitabine, it is a fluoropyrimidine carbamate. It is used mainly in the treatment of colorectal and metastatic breast cancer. capecitabine is well absorbed following oral administration. After being absorbed, it is by itself it is non-toxic, but also it undergoes a similar steps like of dephosphorylation and phosphorylation, and at the end it is converted or hydrolyzed into 5-FU. This step is catalyzed by thymidine phosphorylase, an enzyme that is concentrated in hugely in tumors. So this leads to increased concentrations of the active metabolite of capecitabine in the cancers, and it acts finally in a similar mechanism to 5-FU. So in general, both drugs are causing inhibition of thymidylase synthase, and this leads to cell death due to thymidine deprivation. And as we said, this drug is mainly used for colorectal and metastatic breast cancer. Then we have cytarabine. And cytarabine, it is an analog of 2-deoxycytidine, in which it is not very important to memorize these chemical structures or what they are mean necessarily. What we are talking here, it is mainly or generally related if the drug, if it is related to pyrimidine or purine, or what is the exact mechanism of each drug. So it is important to focus on this more than on the chemical structures. So in this drug, it is an analog of deoxycytidine, and in which the natural ribose residue is replaced or changed to arabinoids. The major clinical use of this drug, it can be used for AML, acute non-lymphocytic leukemia or myelogenous leukemia. Cytarabine enters the cells by a carrier-mediated process similar to what we saw with previous drugs. Like other purine and pyrimidine antagonists that we saw in previous slides, must be sequentially phosphorylated by deoxycytidine kinase, and this is what we saw also with previous drugs, and other nucleotide kinases in order to form an active metabolite, which is called ARA-CTB or cytosine arabinoside triphosphate. ARA-CTB is an effective inhibitor of DNA polymerase, so it prevents DNA elongation due to inhibition of DNA polymerase. And this drug is S-phase specific, so it is cell cycle specific. Pharmacokinetics, cytarabine is not effective when given orally because of deamination of non-cytotoxic to the non-cytotoxic RIU. So here we have a couple of metabolites that we are talking here. So here in the beginning, the active metabolite is ARA-CTB, here RIU, and RIU, it is like inactive metabolites. So that the drug is not given orally, is given intravenously. And it is distributed throughout the body, but it does not enter to the CNS in sufficient amounts. So if there is a brain tumor and there is a need to use this drug, it is recommended to use this drug by intrathecal administration. Cytarabine undergoes extensive oxidation deamination in the body to RIO. This is the same metabolite. This is pharmacologically inactive metabolite, as we said previously. And both of them, the parent drug and RIO, the metabolite, are excreted in the urine. Then we have azacitidine. This drug is pyrimidine nucleoside analog, so it is pyrimidine antimetabolite. And it is used in the treatment of the myeloid dysplastic syndromes and AML. Azacitidine undergoes activation to the nucleotide metabolite azacitidine triphosphate. Just notice here this one in the spelling is different from the parent drug. And this one is the active metabolite. This one is incorporated into RNA and inhibits RNA processing and function, so there is no more transcription to proteins. And this drug is more specific to the S-phase cell cycle. So it is important to notice that the active also, the majority or many of these drugs that are that have anti-metabolites effect should be converted to another metabolite, and these metabolites are active and causing the cytotoxic effects. Gemcitabine, this drug is an analog of nucleoside deoxycytidine. Is most commonly used for pancreatic cancer and non-small cell lung cancer. Gemcitabine is also a substrate of this deoxycytidine kinase, and this is what we saw with previous drugs. And then after that, activation is gonna be to convert it into the active metabolite to difluorodeoxycytidine triphosphate, as we will see here in this figure right here. And this one inhibits DNA synthesis. This drug is administered by intravenous administration, and also it goes and deamination to inactive metabolite difluorodeoxyuridine, and this one is not cytotoxic, it's inactive metabolite excreted in the urine. So here we done with the anti-metabolites. Then now we will start with the antibiotics. The antibiotics also, we have many drugs in this class, but we will talk about them and we will summarize the information as much as we can about the drugs in this class. So the antibiotics mainly cause their cytotoxic effect through interaction with the DNA and leading to disruption of DNA function. The intercalation or this intercalation effect or the alkylation effect with the DNA is not the only mechanisms that's responsible for the cytotoxic effect of this group, but also we have other effects such as inhibition of topoisomerase 1 and 2, and these are very important enzymes for DNA elongation. And also there is a kind of increased the production of free radicals, and the free radicals could cause toxic effect to the cells through lipid peroxidation or other mechanisms. So mainly they are working by three mechanisms: interaction with the DNA, inhibition, topoisomerases, and increase the production of free radicals. So in the beginning, we have anthracyclines, and in this group, we have doxorubicin, daunorubicin, hydrobicin, and ipirubicin with the metoxantrone. Doxorubicin and daunorubicin, both of them anthracycline antibiotics, and this is very clear from the class of this drug. Doxorubicin is the hydroxylated analog of daunorubicin, so they are structurally related drugs. Hydrobicin is also another derivative, and also in addition to other members. These drugs are now used for different uses or different cancers. Even they have structural similarities, and they have structural also mechanism of action or similar mechanism of action. Doxorubicin is the most important one in this class. It is very commonly used cytotoxic drugs for different kinds of cancers, such as sarcomas, breast cancer, and acute lymphocytic leukemia, lymphoma, etc. It is a very popular drug in cancer. Daunorubicin and hydrobicin are now used in the acute leukemias, while with the metoxantrone is used mainly for prostate cancer. The mechanism of action, as we said in the beginning with this class, the doxorubicin, let's say as an example for this group, there is a generation for free radicals, as we can see here in this figure. Like doxorubicin is converted to a reduced metabolite, and this causes a release for the electrons, and electrons are responsible for generation of the free radicals, superoxide ions, hydrogen peroxide, and these ones are responsible for the DNA scission or breakage, and also they are responsible for the for the membrane lipid peroxidations and direct oxidation of purine or pyrimidine bases such as thiols and amines. So in general, these drugs are generating free radicals or oxygen reactive species, and these ones are responsible for the breakage of the DNA strands. In the pharmacokinetics, these agents must be given intravenously because they are inactivated in the gastrointestinal tract. Extravasation after intravenous administration, it is a serious problem. It is a serious problem and could lead to tissue necrosis. Anthracycline antibiotics bind to plasma proteins as well as to many other proteins around the body. So it is widely distributed due to the volume of distribution. They do not penetrate a blood-brain barrier or testes. These agents undergo extensive hepatic metabolism, so it is better given by intravenous administration. Biliary excretion is the major route of elimination because these drugs could cause like a side effect, a kind of cosmetic side effect. It is a dark red color or discoloration on the site of the injection, and this causes that visible blood vessels around the site of the injection or the infusions. And also they could lead to rediscoloration of the urine. Adverse effect, the most important side effect related to these drugs is the cardiotoxicity, which is a reversible dose-dependent side effect. It is a very serious side effect. And it is very common with the doxorubicin, but I think it is less likely to happen or with lower incidence with idarubicin and epirubicin. Cardiotoxicity apparently related to the generation of free radicals and lipid peroxidation. And this is what we saw from the mechanism of action through generation of reactive oxygen species. Co-administration of monoclonal antibody trastuzumab, trastuzumab with the doxorubicin or epirubicin increases the incidence of cardiotoxicity and congestive heart failure. Protective strategies. There we have two protective strategies here. We have to use the iron chelator agent, dexarazoxane. This one has alkylating activities and could decrease the level of the drug in the heart. And also there is another protective mechanism by using liposol dosage form. So using liposomes to encapsulate the drug and to prevent the drugs from the direct contact with the cardiac cells, such as cardiomyocytes. Then we have bleomycin. And bleomycin is a mixture of different glycopeptides. Like anthracyclines antibiotics, it causes scission or scission of DNA by an oxidative process, also through generation of reactive oxygen species. Bleomycin is cell cycle specific, and this is the only exception. Most of them, they are this class, all of them are cell cycle nerve specific, except bleomycin. It is cell cycle specific and cause cells to accumulate in G2 phase. Is used mainly to treat testicular cancers and Hodgkin's lymphoma. The mechanism of action is similar to what we saw on the previous drugs, that DNA bleomycin ferrous complex undergoes oxidation to bleomycin ferric complex. And this process causes to liberation of electron ions and electrons react with the oxygen and generate superoxide and hydroxyl radicals, and both of them reactive oxygen species, and these ones are attacking the phosphodiester bonds of the DNA, resulting in the DNA scission. And of course, there is aberrations of chromosomal due to the breakage of DNA strands. And this is the mechanism of action, just in the figure, the same that we talked here. Pharmacokinetics. Bleomycin is administered by a number of routes. Bleomycin inactivating enzyme hydrolase is high in number of tissues, but it is very low or sometimes absent in the lung and the skin. And this is the major reason for the toxic effect or side effects of this drug in these two organs. Most of the parent drug is excreted and changed in the urine, and there is a necessary dose adjustment if the patients have renal failure. Adverse effect, the most common and serious side effect with the bleomycin is the pulmonary toxicity. And this causes certain symptoms such as rales, which is abnormal sounds from the lung, cough, infiltrate to the potentially fatal fibrosis. And this pulmonary fibrosis is called bleomycin lung. Also, patients could suffer from hypertrophic skin changes, and this is what we talked about, that the hydrolase enzyme, it is very low or absent in the skin and the lung, so the side effects are concentrated, is a kind of concentrated in the skin and lung. Hyperpigmentations of bleomycin. Also, it is noticed that this drug is rarely caused myelosuppression. Then we have here the after antibiotics, we have the alkylating agents. And in this class, mainly their effects or their cytotoxic effects is related to the binding to the nucleophilic groups in various cell constituents, such as DNA, RNA, or other nucleophilic groups inside the cells. Alkylation of DNA is the most, is the most important step in the cytotoxic mechanism or action that is induced by these drugs. Alkylating agents do not differentiate between the cells which are in the cycling or they are resting cells. So they are attacking both dividing and non-dividing cells. However, their effects are most toxic to the rapidly dividing cells. Can be used in combination with other agents from other classes. In addition to all these drugs in general could cause a common side effect, mutagenicity or carcinogenic effect, and this is called like secondary malignancies such as acute leukemia, and can be treated by anti-cancer drugs. So here we have cyclophosphamide and ifosfamide. These drugs are closely related mustard agents, and they have a very similar mechanism of actions and side effect profiles. The cytotoxic effect happens after generation their alkylating species, so they must be transformed into other active metabolites, and these metabolites have the alkylating activities, which are produced through hydroxylation by cytochrome 450, and these agents have a broad clinical spectrum and can be used as a single agent or in combination, as we said in the beginning or in the previous slide, to treat various neoplastic diseases such as Hodgkin lymphoma, sarcoma, and breast cancer. So with the mechanism of action, cyclophosphamide is the most commonly used one in the alkylating agents in general. Both cyclophosphamide and ifosfamide are firstly biotransformed into hydroxylated intermediates, and this happens in the liver by the cytochrome 450. And this hydroxylated intermediate undergoes metabolism to form an active compounds, phosphoramide, mustard, and acrolein, as we can see here in the figure here. So in the first step, there is a hydroxylation in by the cytochrome 450, and then after that, there is another step of the metabolism and converted the hydroxylated metabolite into the phosphoramide, mustard metabolite, and acrolein. And these ones are responsible for the cytotoxic effect of these drugs through a reaction directly to the DNA alkylating effect on the DNA. Pharmacokinetics. Cyclophosphamide is available as oral or IV preparations, while the ifosfamide is only available for IV administration. Cyclophosphamide is metabolized in the liver into active and inactive metabolites, and minimal amounts are excreted as a parent drug and change the drug, while ifosfamide is metabolized by cytochrome 450, and it is mainly renally excreted. Adverse effect. The most common and unique side effects with these drugs is the hemorrhagic cystitis, and this could lead to fibrosis of the bladder. And bladder toxicity, it is related to the metabolites of these drugs. So in case of cyclophosphamide, it is related mainly to acrolein, while there are other toxic metabolites are related to ifosfamide. So these are due to the toxic metabolites that are generated from these drugs. As a protective mechanism that are now used, we have to use, let's say, or tell or advise the patients for adequate hydration. And also there is another way by IV injection of mesna and mesna-like alkylating agents that are responsible to decrease the concentration of the drug inside the urinary tract. But it is a protective mechanisms, but I think they are not completely protective. Still, this side effect happens with these two drugs. Neurotoxicity, it is also reported with ifosfamide, and maybe it is related to another toxic metabolite, chloroacetaldehyde. Then we have nitrose ureas, and here we have two drugs, carmostine and lomustine, and also these are closely related drugs. They have similar structures and similar mechanism of actions. However, there is an important point here or advantage for using these drugs, their ability to penetrate the CNS, so they are commonly used in the treatment of brain tumors. Nitrose ureas exert or show their cytotoxic effect also by alkylations, and this alkylating effect inhibits replications and RNA and protein generation, alkylating effects to the DNA, and this inhibits transcription of DNA to RNA, and this inhibits transcription RNA to proteins. Although they alkylate DNA in resting cells, but it is cytotoxic effect. It is also they exert a cytotoxic effects in actively dividing cells. So they are working on both cells in the resting phase or actively dividing phase. Therefore, non-dividing cells, it could be, let's say, that the cells which are actively dividing are more susceptible to these drugs more than the cells in the resting phase, due to the ability of the cells in this phase to repair their DNAs after this disruption. Also, there are other effects are also related to nitrose ureas, such as inhibiting other enzymes that are responsible for the protein generations, and such as the carbomylation of amino acids and proteins in the target cells. Pharmacokinetics, carmostine is used by intravenous administration and wafer implants, while lomustine is given orally. Because of their lipophilicity, as we said in the beginning, these agents distributed widely around the body, and their ability to penetrate CNS, it is very good capacity. The drugs are extensively metabolized, and they are excreted mainly by the renal excretion. Then we have dacarbazine and temozolomide. And these drugs also alkylating agents, but also they need biotransformation into active metabolites. And in this case, we have this metabolite, methyl triazinoimidazole, carboxamide, MTIAC. And this metabolite is responsible for the alkylating activity of these two agents through forming a methyl carbonium ions that attack the nucleophilic groups in the DNA molecules. So these two drugs have been transformed into MTIC metabolite, and this metabolite will be transformed into methyl carbonium ions, and these ions are attacking the DNA molecules or the nucleophilic groups in the DNA. The cytotoxic action of dacarbazine has been mainly attributed to its ability to attack or alkylate DNA in certain sites, and in this case, with the dacarbazine and the O6 position of guanine, and this leads in general to methylation of DNA. And methylation DNA decreases a lot the ability or the capacity of DNA to transcript itself to RNA. So they are attacking or targeting certain sites in the DNA, such as in the case of dacarbazine O6 position of guanine. Dacarbazine now is used to treatment various cancers such as melanoma and Hodgkin's lymphoma. Temozolomide also is, as we said in the beginning, is related structurally to dacarbazine. Both of them, as we said, have to be transformed to this active metabolite, MTIAC, and this one is responsible for the methylation of DNA. However, temozolomide does not require cytochrome 450 for this transformation. It is easily transformed just only in the normal physiological pH. Then, temozolomide inhibits other enzymes, not only just methylation of certain sites in the DNA, but also inhibits other enzymes such as O6 guanine DNA alkyl transferase. Another difference in temozolomide in comparison with dacarbazine, these drugs have the ability to cross the blood-brain barrier, so it can be used in the treatment of the brain tumors such as glioblastomas and astrocytomas. It is also used for the treatment of metastatic melanoma. Temozolomide is administered intravenously or orally and has an excellent bioavailability after oral administration. The parent drug and metabolites are excreted in the urine, as we can see from this figure. So here we will stop at this point, and we will continue in the coming lecture with the other alkylating agents. Thank you very much. And of course, if you have any questions, just let me know and in comments, and I will answer these questions. Thank you very much.